Hello everyone. So welcome to my channel, and now we are going to start with the new unit, and that is genetics. In that we are going to study first chapter, that is principles of inheritance. And I hope you know this man. He is Gregor Mendel, and he was in the 19th century, and his parents were what? He was from a very small family. They were farmers, isn't it? And since he was in that background of village, and therefore he was knowing about growing of plants and keeping of animals, all these things. Okay, at the age of 21, Mendel left the farm and entered a Catholic monastery in the city of Brunn, that is now known as Brno, in which is in Czechoslovakia. In 1847, he was ordained as a priest, adopting his clerical name. Then he was given Gregor. He subsequently taught at the local high school, taking time out between 1851 and in 1853 he went out to study in the University of Vienna. Okay, after entering to Bruno, he re-entered his life as a priest, as a monk, teaching monk, and began the experiments that eventually made him famous. Mendel performed experiments with several species of garden plants, and he even tried some experiments with honeybees. His greatest success, however, was with, as all of you know, it was the pea plant, that is Pisum sativum. He completed his experiments with peas in the year 1864, or you can say in, and he published it. Basically, he completed in 1864 his work, 1864, and it was published in 1865. Okay, so this was with Gregor Mendel. And then his in History Society of London, it was published there. And unfortunately the paper didn't got very famous, and it was until in 1900 it was rediscovered by three scientists. I hope you people know. It was Hugo de Varies, Carl Scorens, and von Tischer-Mark, so from Austria. Okay, so this is a brief history we should know about, and we should salute these people. Especially I salute this person. Why? Because uh, he was doing experiment, and he was a mathematician as well, and he applied his mathematical study to a biological experiment, which is something very unique. And it gives na and therefore he was able to look at his experiments in a very different way. And when you will also see his experiments, how simple experiments, but deduce certain conclusions, which is really fantastic. Okay, so I just salute this man, and we should learn from them a great deal. So let's come to this word genetics. Okay, genetics means what? In Greek it means to generate. Okay, it means to generate, and it is what? It's the science, no, where we not only, no, if you look at around in your area or in your environment, what do you find? There are different kind of people, and even within your family, if you look at, what do you find? Are you similar to you exactly like your parent? No, you are dissimilar. So you don't find ki you are exactly like your parent, na? Maybe your siblings are also different. Maybe some has got brown eyes, some have got black eyes. It varies, isn't it? So why is it varies? So genetics is the study where we study what transmission of genes, and as well as what the expression of the genes. Like I have inherited black here, isn't it, from my parents, and how it expressed in my body, that is what we talk about now expression. Now, how that gene which get transmitted from one individual to another, how it expresses itself also. Okay, so genetics involves both the things, and here we study both similarities as well as variations. It's nothing like we are going to study only one thing. It's not that way. We study both genetic similarities as well as variations. We study the differences between parents as well as the offspring. Okay, or among the individuals of the population. Even when we do a population study also we do in genetics. Okay, so there are many facets to it, and as we will keep on moving toward you, it will be just unfolded to you. Okay, now moving on to this heredity, where it is what the transmission of the genetic information from parent to offspring. Okay, and it generally follows predictable patterns. Okay, and you can see here when you study na, so you will be able to predict it. We will deal with those cases also, and I will also give you now certain questions based on it also, where you do it, because until and unless you do it you won't be able to predict. So there is a general rule that follows by which we can predict what could be the pattern. Okay, in diverse organisms like humans, penguins, all the organisms, because these principles which Mendel has laid down, it's now applicable to all the organisms, because the rule the genes they follow is same in all the organisms, isn't it? So let's move on. Let's see the Mendel's experiment. First of all, Mendel, as you know, he has worked on P. I've already talked about it, and the major pea plant which we study for, on which he did study, is Pisum sativum. Okay, so it is Pisum sativum. Okay, it is Pisum sativum. Now what do we find? That is the technical name. Okay, now he basically, the what is the characteristic of this pea plant? This pea plant it's a bisexual flower. First of all, it's bisexual. Okay, so what do you find in it? That it has the property of self-fertilization, self fertilization, as well as it has the capacity of cross fertilization. Now you can easily cross fertilize it also. Okay, so it is self fertilization also, it happens in it, and basically it is self fertilized in nature, but you can cross fertilize it also. Okay, so that was the basic thing which Mendel took this plant. Why? Because he found that this plant showed distinct observable characters to him. If I will talk about it, there are discrete characters we talk about, and there are continuous characters. So one by one, as we will keep on moving, we will talk about it. So these are discrete characters. What kind of characters they are? They are discrete. Why we say them discrete? Because they are either this or that. It's nothing like in continuous kind of variation, like you find height of a person or skin color of a person. What do you find in that? You find that it varies. In a population, if you look at, you can innumerable of heights, innumerable of skin color, but here it was only two type of traits were seen. Okay, either they were tall or short. Okay, so let's look at either the flower color was what, purple or white. Okay, either the pea shape was what, round or wrinkled. Pea color was what, yellow or green. Okay, so it was nothing shades of we are finding. No, we are not finding in the nature shades of, just two types. Therefore we are calling them as discrete. Okay, either pod color is what, green or yellow. Okay, pod shape is what, Pod shape is what? Inflated or constricted. Plant height is what? Tall or short. Flower position is what? Axial or terminal. Okay. So these were the 7 characters which was studied by Mendel. And remember, character is what? It's an observable. See, I've just written it also for you where it can be highlighted that it's a character which is observable. Okay, it's an observable character. You know? So the things which you can look at it, you can observe at it. Okay, that is called. And what is trait? Try to understand, because these words will come and you should know the difference between them. So it's a particular form of a character. Like if I say purple, this is one trait. This is one trait. This is one trait, round, yellow, wrinkled, another trait. So heritable trait is passed from parent to the offspring. So what is a heritable trait? It's a trait which passes from your parent to your offspring, is called as a heritable trait. Okay, fine. So I hope you know by now it's clear what is the character. Character is the observable feature. Like I talk about height is one character, but the trait is what? Whether it is what? Short or tall. Okay, is that clear? Now let's move on to the Mendel's first experiment that he did, and it is called as what? It is called as Mendel's experiment, monohybrid cross. We call it as what? We call it as monohybrid cross. As you can see here, I'm just highlighting for you. It's called as monohybrid cross. Now coming back, what did Mendel do? First, now he took tall plants, and first I will try to introduce you one thing which is called as pure line breed. You should understand what is pure line breed. Okay, what is pure line breed? Pure line breed. Pure line breed is what? Where now or what he did basically, that he now self-fertilized the organisms for many generations. Like he self-fertilized tall plants for many generations. So now he is very sure that this plant is going to give me a tall plant. So similarly, he self-fertilized the short plant for many generations, and he is very sure that it is going to give what? Short plant. So he took pure line breed plants, and in that he took what? Tall plant, tall pea plant, and he took what? Short plant. And they were pure line breed, remember. And this is what is your parental generation. Okay. Now what he did, he cross fertilized them. Okay, I'm reiterating it. What he did, he cross fertilized them, and what did he saw that fertilization, he got all tall plants, and this generation is called as F1 generation. Why F we write? F is written for filial, which means what? Sons and daughters. Okay, generation. Okay, so he took your he found that were all tall. In F1 generation, he found that all were tall. Now what he did, he allowed these organisms to, F1 organisms, to self-fertilize. As I have already told you, both can be done in these plants. Self-fertilization. Okay, self-fertilization. Now what is happening after doing self-fertilization? To his surprise, what he got? He got two kind of plants. So what were they? They were your, when he self-fertilized, he got tall plants, and he also got dwarf plants or short plants, you can say. Short, dwarf or short, same thing. So what did he got? He got tall and dwarf, and when they calculated, see I told you, use mathematics. So he counted the individuals and he calculated their ratio. It was in the ratio of 3:1. Now, from the simple experiment, now Mendel put forward certain laws. Okay, so I will come back to that law and what made him say that these kinds of law, that also I will talk about. Okay, now uh, what happened, like earlier, no, this was the notion, earlier this was the thinking that keep basically the organisms, no, they thought that this was the earlier notion, and in scientific society also it's very difficult, like in our day-to-day life, even if you see people are highly conditioned, and to break that notion, break that belief is very difficult. Similar is with the scientific no, people also. So they were under the notion that the genes they blend. They thought that the genes they blend. But by looking at this experiment, Mendel said no, genes do not blend. What made him say so? That genes never blend. Okay, that made him say so because when he, there were the two pointers, look at it carefully. When he cross-fertilized tall and short, according to the blending's principle, he should get something intermediate here. Something intermediate height of organism, but he didn't get. Okay, and also in the second generation, this is called as F2 generation. What did he get? He got this dwarf organism. And by looking at this dwarf organism, he said, had blending happened, if blending would have happened, then I would have never got this dwarf plant. Example I'm giving you, like if I mix color, suppose I mix red and white, will I be able to get white later on blending? No, you can never get that. So he just used his logic and said, how can I get if blending would have happened? Therefore, he put forward that genes are what? Genes are particulate matter, or discrete units. Okay, so very simple experiment, but coming to a very great kind of concepts, and from this he came to his laws as well. Okay, so no, this is your what I have written, tall, short, tall, this is showing you what the phenotype of the organism, the physical appearance of the organism, how the organism is appearing. Okay, कैसा दिख रहा है organism, है ना? So this is showing you what this the physical appearance of the organism, isn't it? I have written the physical appearance of the organism, and technically we call it as what? Phenotype. So I hope this word is clear. Phenotype is the physical appearance of the organism. And here what did he observe? This ratio, if you look at, it is what he said, that the phenotypic ratio of the F2 generation is in monohybrid cross, it is 3:1. Now, how to, there is another term which I should tell you here, that is what? That is your genotype. Now try to understand both the terms. Okay, so what is genotype? So, genotype. Genotype is the genetic composition. Okay, genotype is what? What is the genetic composition? So to express the genotype, he used the letters. Okay, he used alphabetical letters because according to him they are particulate matters. So he said, okay, let me put some letters for these genotype. Okay, so he said what he did, he put for dominant character. Now what is dominant character, I will tell you. What he did, he took tall, because the one that expressed in F1 generation is called as what, dominant. Okay, the after parental generation, the one that expresses himself in F1 generation, he called it as dominant. Okay, so based on that, what he did, he put the tall ones, he represented it with, I will just change the color for you people. What did he do, he just put capital T for that, for the dominant character, and for the trait which is short, he used what, small t. Okay, now another term I will introduce, because there is a lot of terms here which will come to you, we are studying for the first time. So allele. Now what is allele? Allele is what? Allele is the contrasting form of a gene. Okay, is a contrasting forms of a character. Okay, also you can say. So allele is what? Is a contrasting form of a gene. Suppose the gene is what? Height of the plant. We are talking about the gene height of the plant, and it has got two alleles. So allele is what? Contrasting. Contrasting forms of a gene or a character, you can say. Okay, so that is your allele. Clear? So he said he expressed the dominant allele with capital T and the recessive allele with the short t. Okay, or you can say he expressed tall with capital T's and short with small t's. Clear? Recessive and dominant we will come. Okay, now in this case, now you have to make what? Your gametes. What will come? You know meiosis will happen in sexual reproduction. Okay, you have read in class 11th meiosis, so you know that gametes will be formed, and you know that gametes contain only one what, homologue, of the homologous chromosomes, only one chromosomes. Okay, so it will have only what, one T. Therefore gamete will have only one T. Okay, and this will represent only one T. Okay, so now what happened, now what he did, just let me use this. Okay, this is small t. Okay, now on fertilization, what should I get? I should get T and small t. Okay, now again you have to make the gametes. Okay, and you have to form the individuals. Okay, so let's make the gamete and form the individuals. Okay, so if I have got this, if I have to make the gametes, I won't take another page, because then I have to make it again. So I'm doing it here only for you. Just you can see here, if I have to go with this one TT. Okay, so there will be two kinds of this T and this small t. I'm just separating it out, okay, for you. Now if I have to now self fertilize it, means I should have a male and a female, both, isn't it? Because two individuals you need for fertilization, even if anthers and pollens you need for that. So suppose this is your male and this is your female gamete. Okay, so this is capital T, this is capital T, small t. Okay, this is capital T, small t. Okay, and this is small t, small t. What do you have to do? You have to just multiply it with this. So capital T, small t. Okay, small t, capital T, and TT. So now what do you get? What did he find? He found that the three were tall. So these three are tall. These three are what? They are tall. And this is short. Okay. So if I have to put the genotypic form, so what will I put? TT, then 2TT and what? Small tt. So what ratio I am getting? I am getting 1 is to 2 is to 1. So this is called your genotypic ratio. So the genotypic ratio of the monohybrid cross is 1 is to 2 is to 1, and the phenotypic ratio is how much? It is 3 is to 1. And remember, the one that expresses itself in, again I am repeating for you, the one, the allele, okay. So I will just rub few things so that I write for you. The allele that expresses itself, write down all of you, the allele that expresses itself, okay, itself in F1 generation or in heterozygous condition is called dominant allele. Now why I am saying so? It's called dominant allele. Okay, you can see here tall is expressing itself in F1 generation and it is expressing in heterozygous. Heterozygous means one is a dominant allele and another allele is a recessive allele. Okay, so you can see here one is here this kind of individuals, okay, with both capital T's or both small t's, we call them as what? We call them as homozygous. What do we call them as? We call them as homo means same, zygous. Okay, we call them as homozygous, and such kind of individuals we call them as what? Heterozygous, because the alleles are different. Okay, so he said that the alleles that expresses itself in F1 generation or in heterozygous condition, तो इस condition में कौन अपने आप को express कर रहा है? Tall. Plant क्या है? Tall है. तो capital T is able to express itself, not the small t. Okay, therefore he said it's a dominant allele. And now write down what is recessive allele. What did he say? He said the allele that expresses itself, okay, expresses itself in homozygous, only expresses itself only in homozygous condition is called recessive allele. Okay, so these were some basic terminologies and a very simple experiment conducted by Mendel, but very effective. Okay, so based on this cross, if you look at here, I have also written for you, it's a monohybrid cross. Why I am saying it's a monohybrid cross? Because mono means single, and since we are doing cross fertilization, so it's a hybrid. Okay, we are choosing two different plants, so it's a hybrid. Okay, so monohybrid, single character. So monohybrid cross is a cross where we study the inheritance of a single type of character. So we have taken a single character, that is height of the plant, and we are studying its inheritance. Okay, so this is called as inheritance of one gene or monohybrid cross. Okay, now this figure is showing you what? Ki Mendel did this monohybrid cross with all the 7 characters. And with all the 7 characters, on the basis of this result, he said, okay, this is dominant, this is recessive. Okay, and look at the numbers he counted and the ratio he got. So in all the ratio For the dominant and the recessive, he got in the ratio of what? 3:1. He got in the ratio of 3:1. Okay. So with all the 7 he did and came to this generalized what? Conclusion. Okay. Now we will come to let's come to the conclusions drawn by the Mendel. Already I have told you, but still I will just write or put it into words for you. Okay, so what was the first conclusion drawn by Mendel? That the characters are controlled by genes. Okay, characters are controlled by genes. This was the thing. Okay, and he said that genes are discrete unit. Okay, he said that genes are let me take another pen so that's visible. He said that the genes are discrete unit, units. Okay, never blend, they never blend. So this was the important conclusion drawn by your Mendel. Then the another was that genes occur in pair. Okay, what did he say? What did he say? He said that genes occur in pair in an individual. Okay, occur in pair in an individual. Okay, this was his day, and these genes, genes segregate. He said that genes segregate during gamete formation. What did he say? Genes occur in pair in an individual. Individual में वो pair में होते हैं ना? Like individual has got what? It has got TT, है ना? If you remember with the TT, and the gamete has got only single, whether it is capital or small t. Okay, depending upon the individual, the gamete will be. If it has TT, then the gamete will have only T, capital T. If it has capital T and heterozygous, then it has got both the. It could be capital T and small t, both. And the third thing, he said that the allele expresses itself in hybrid or in heterozygous condition is called as the dominant allele, and the allele that and the hybridization allele, the hidden allele, you can say, is called as what? The third point that he gave that the allele that expresses is dominant, that expresses in F1 generation as dominant allele, he said, or in heterozygous condition. Okay, you can write down all allele that expresses in F1 generation or in heterozygous condition is a dominant allele, and the hidden allele, and you can write, and the hidden allele is recessive. This was his conclusions, and based on these conclusions he gave 2 laws. What was the Mendel's first law? Don't forget. What is the first law? देखिए कई बार क्वेश्चंस पूछता है, बच्चों को नहीं पता होता which is the first law. The first law is law of dominance. Okay, and this law says that what does it says? What does this law says? That we have put forward 2 laws were put forward. Okay, what does this law says? That the allele that expresses itself in heterozygous condition or in F1 generation is a dominant allele. This is a law of dominance. And the second law was your law of segregation. Remember, law of segregation, or you can also call it as purity of gametes. And it's what kind of law? It's a universal law. This is an universal law, remember one thing. And this law states that what? It states that the gametes are pure for the gene, for the allele, and the another way you can put it as that the alleles segregate during gamete formation, like capital TT separate or capital T small t separate during gamete formation. Okay, let me explain you. Let me go back to meiosis and explain you this thing. Now you can see here that this is the picture which is showing you chromosomes and meiosis. Okay, and it is also kept here, the chromosomes, the alleles have been kept in the chromosome. You know that the genes are located where? On the chromosome. So in this picture I am showing you that this is your what? This is your homologous chromosome, as you can see, and you can see this is capital T and this is small t. Okay, so one chromosome has got small t and another chromosome has got capital T. Okay, so this is F1 generation, I can say. Okay, from your what you have read right now. And here you can see these are homologous chromosomes. Why we call them as homologs? Because they homologous chromosomes are what? They are same in the shape, size and the location of genes they carry. Look, capital T are placed here and even small t's are in another chromosome. It's not that capital T is here and the small t has been placed here. No, the position is also same. Okay, now in meiosis 1, what is happening? First thing is happening is your homologous chromosomes separate. Homologous chromosomes separate, isn't it? They separate out. So homologs are separating, and now in the second meiosis 2, what is happening is your sister chromatids. Your sister chromatids separate. And as a result you can see you are getting these gametes. Okay, so what are these forms is your gametes. Now gametes are what? Well, that's why I'm saying they are pure for the genes, because they are containing only capital T or only small t. Or you can say that the alleles segregate. Now they have segregated, capital T and small t has segregated. Okay, so now I have jotted down for you people all the definitions so that it helps you. Okay, and you can go through it once. Already I have dealt with all these kinds of terminologies with you, and you can go through it. Okay, now coming back to one of the major thing is your test cross. Okay, so coming back to your test cross. Now, in F2 generation, if you look at, we got not dominant plants, okay, and but their genotype was of two type, isn't it? One was this and one was this. Now both appeared what? Tall. Now, how do one knows? How do one knows whether they are homozygous dominant? Whether this homozygous dominant or heterozygous dominant? How do one knows? कैसे जाने? कैसे पहचाने? Okay, for that what has been designed is your Mendel did a test cross. What was he did? Test cross is a cross, remember, where the now dominant plants are now, because for dominant only you don't know the genotype, they are crossed with the recessive parent. Remember, they are always crossed with their recessive parent. Okay, so suppose this is the case. Now the cross is going to be made with the recessive parent. Now all the individuals, let me make this. What do you get? All the individuals in this case you will get what? Are tall. What do you get? This is the forked method. That was a Punnett square method I showed you earlier. So they are all tall. When you get this all tall, then you can say that the individual is homozygous dominant. Now, taking the another case where we are having heterozygous individual, where they are what? Capital T small t into the dwarf parent. No? Dwarf parent. Okay, recessive parent. Now you just try to cross them. what do you get? You will get the ratio of 1:1. So you get the ratio of 1:1, that is tall is to dwarf. So when you get the ratio of 1:1, that is 1:1 is tall to dwarf, then what do you say? That they are what? Heterozygous dominant. Okay. So I have covered today, giving you some terminologies and monohybrid cross and two laws of Mendel. Okay. And very important concepts I have dealt today. So please go through it, because this is the basics. Okay. Once basics is strong now, you can make a high building. Okay. With this, okay, I will say goodbye and God bless you all, but don't forget to subscribe.